Hi, my name is William, and I'll be presenting my Journal Club article on geographical relationships of the microbiome of reefs located in the Pacific. This research was conducted in 2023, and the article is called Diversity of the Pacific Ocean Coral Reef Microbiome. The Bluff. This study attempts the vast microbiodiversity associated with coral reefs and emphasizes the importance of considering geographic and environmental factors in coral microbiome research. These insights are vital for incoming conservation and management strategies aimed at preserving coral reef ecosystems. Now, for the background, coral reefs are complex ecosystems where the health and function of coral reefs are intricately linked to their associate microbiome communities, collectively known as the coral microbiome. Understanding diversity and distribution of these microbiome communities is essential for assessing coral resilience and ecosystem stability. With increasing environmental pressures such as climate change and pollution threatening coral reefs globally, there is an urgent need to elucidate the factors shaping microbiomes to inform conservation and restoration efforts. Methodology. This study utilizes 16S rRNA gene meta-barcoding to profile bacterial communities associated with three coral genera: the Millipora, the Porites, and Poeciloporas. Samples were collected from various locations across the Pacific, and high throughput sequencing was employed to assess the microbial diversity and composition. I found these two articles in 2002 that would really kind of lay a foundation for a better understanding of some of the nuances of understanding the reef microbiome. The first is diversity and distribution of coral reefs associated bacteria, which this study it analyzes over 1,000 bacteria, their DNA sequences from three massive coral reefs down in Panama, Bermuda, and revealed 430 distinct bacterial ribotypes with projections suggesting up to 6,000 ribotypes upon further sequencing. Notably, half of these sequences shared less than 93% identity known 16X sequences. This is telling us that there's a whole heck of a lot of unknown when you start hopping in the ocean and looking at reef microbiome, which is also a good thing, but also can be a limiting factor, and I'll explain further. Additionally, I chose the article Prokaryotic Diversity and Its Limits. It kind of touches on what I was previously talking about. Estimating the total number of prokaryotic species in any environment is challenging. However, by assuming that species abundance follow a log normal distribution, it is possible to estimate species richness using the ratio of total numbers of individuals to the abundance of common species, leading to a preliminary estimation such as 160 species per milliliter in the ocean. Obviously, they're not measuring individual microbiome aspects. They're drawing statistical analysis and making confident decisions. Coral reefs are among the most diverse ecosystems on Earth. For this research study, supporting a vast array of multicellular organisms that rely heavily on associated microorganisms for health and nutrition, a comprehensive study systematically sampled 3 coral morphotypes, 2 fish species, and plankton communities across 99 reefs from 32 islands in the Pacific Ocean to assess reef microbiome composition and geography. The findings reveal immense richness of reef microorganisms compared to other environments, suggesting that the total prokaryotic diversity in the coral reefs approximate current estimates for the entire Earth. Now, this is going to come with some obvious advantages and disadvantages. Thankfully, this study that I chose has a large database, which allows them to make a more clear picture of what exactly is happening with the reefs and how it's functioning. The study also, they were able to reveal how high microbiome diversity with that data and the insight and gain an insight to biogeographical patterns. Now, it's got its disadvantages and limitations, if you prefer. So there's like a temporal variability. What that means is it's like a picture. So you take a picture of it, and that's how things were when you last looked at it. So obviously nothing is staying still in the environment. So what we may know right now may not be right or the correct actions or what's currently happening. So then we have functional analysis, which was kind of what I was getting into. And then lastly, another disadvantage is host-specific interactions. Again, we don't have everything sequenced and everything figured out and how everything goes together. And this plays roles that we don't quite understand yet and when it comes to the reef microbiome. Figure 1b, this panel shows a number of different microbiome species. Richness increases as more samples are collected. The curve that you see over here kind of goes up, then it starts just tapering off slower and slower. So the curve depicts the cumulative count of unique microbial species identified as sampling continues. Now, I want you to imagine you're collecting samples from the ocean and you identify different microbe species. Initially, with each new species you discover, over time, as you gather more samples, the rate of discovery of new species slows down because you've already identified most of them. So that kind of gives you an idea of why the curve is in the shape that it's in. Now, to better discuss what figure 1c is, this panel presents a Shannon Diversity Index. It's a measure that accounts for both the number of species, their relative abundance and community. The box plot displays the mean value, if you're familiar with statistics, the first and third quartile of the box, and the range, excluding outliers. The whiskers extend up 1.5 times for the interquartile range. This Shannon Diversity Index helps us understand not just how many different microbial species are present in a sample, but also how evenly distributed they are. A higher Shannon Index indicates a more diverse and balanced microbial community. The box plot provides a nice visual summary of these diversities across all collected samples, showing the typical range and variability of microbial diversity in the study. Now you'll see the key at the bottom. It's a mixture of microscopic organisms and fish and reef, and what they're basically doing is they're running genetic testing on the microbiome of these species using what's known as amplicon sequence variants which provides a high-resolution method for identifying unique microbiome sequences, which allows for precision differentiation between closely related microorganisms, such as targeting the 16S rRNA gene in bacteria. Figure 1D, Bray-Curtis-based NMDS ordination. This panel illustrates the difference in the microbiome community composition among various biomes, plankton, coral, fish, using a method called non-metric multidimensional scaling based on Bray-Curtis dissimilarities. The NMDS plot positions samples based on their microbiome composition with a closer point indicating more similar communities. The density plot on the right shows the distribution of MDS2 values for coral, small plankton, large plankton, fish gut, and fish mucus samples. And samples that are closely together have similar microbes, while further apart differ more. The density plot on the right shows how samples from biomes are distributed along the MDS2 axis, helping to visualize the difference and overlap in microbiome composition among these groups. Figure 1E, prevalence and relative abundance of ASV. This panel displays the prevalence and relative abundance of amplicon sequence variants, the ASVS, in plankton, coral, and fish samples. The chart highlights bacterial families. For example, potential seminots are marked in black, and potential pathogens are in gray, and all others annotations in white. By highlighting these bacterial families, researchers can identify which microbes are common or rare in each biome and assess their potential roles. So that's pretty interesting. So they took a look at all this data, and in summary, no clear biogeographical patterns were found, as distinct islands sometimes had similar communities while geographically close ones differed. Both geographic distance and environmental factors, temperature, salinity, pH, phosphate, silicate, influenced microbiome composition, with geography having a strong correlation. Each coral morphotype had a distinct microbiome community. Specific microbiome taxons were found only on certain islands, while others were widespread. A core microbiome was identified, but no microbiome species were present in all samples. Okay. Figure 2, Bray-Curtis-based MDS ordination, showing differences in microbiome community and composition within each biome. So panel A. This panel compares the microbiome community associated with the 3 corals notated below. The MDS plot displays each coral species in a different color, illustrating how similar and different their microbiome communities are. If, for example, points are close together, it means that these have similar microbiome communities, indicating that each coral species hosts a distinct set of microbes. Panel B shows the overall microbiome community composition, focusing on the 10 most abundant bacterial orders found across the 3 coral species. The bar chart displays the relative abundance of these top bacterial orders within each coral species. Think of it as a breakdown of the most common bacterial groups present in each coral species. By comparing these groups, researchers can identify which bacteria are prevalent in each coral type and how their microbiome communities differ. Additionally, panel C, this panel examines the microbiome community found in the gut and the mucus of the two fish. Similar to panel A, this plot shows how microbiome communities cluster based on the sample type in fish species. If gut samples from a particular fish species clusters together and separates from the mucus sample, it indicates a distinct microbiome community in these different habitats within the fish. Now for panel E, this investigates the microbiome community in plankton samples collected from seawater near the islands, the surface of the water over the colonies, and water close to the colonies, and so on and so forth. So the MDS plot compares different water samples to assess variations in the microbiome community. By analyzing how the water sample clusters, researchers can understand how approximate to coral colonies influence the composition of microbiome communities in the surrounding waters. Similar to panel E, this panel examines the microbiome community in large plankton across the same type of water samples. This analysis helps determine whether large plankton organisms have different microbiome communities compared to the smaller ones, and how these communities vary with their proximity to coral colonies. Now overall, Figure 2 collectively illustrates the distinctiveness of microbiome communities across various biomes. Panel A and B revealed to us that each coral species hosts unique microbiome fingerprint. Then panel C showed that the gut and the mucus microbiomes of fish species are actually species specific. And panel D indicated that the corals maintain microbiome communities distinct from nearby free-living plankton. Then panel E and F demonstrated plankton microbiome community compositions vary depending on their proximity to coral colonies and plankton size fractions. These findings shed light on the specificity and the diversity of microbiome association in the marine ecosystem. Now, Figure 3 illustrates the diversity of microbiome communities associated with the 3 coral species across various Pacific Ocean islands, using the Shannon Diversity Index as a measure. Now, the index quantifies both the number of different species, basically the biodiversity and richness, and the evenness of their distribution in the community. The higher values indicate a greater diversity. By examining these figures, researchers can identify patterns and hotspots of microbiome diversity in coral reefs across the Pacific Ocean, which is critical for understanding coral health and ecosystem resilience. Figure 4 examines the factors influencing composition of coral microbiomes across the Pacific Ocean by analyzing pairwise dissimilarities in microbiome communities associated with the 3 coral species. Pairwise dissimilarities measure how different 2 microbiome communities are from each other. In this figure, it is calculating using Euclidean distance, where larger values indicate greater differences between communities. Now, what does this mean? Think of it like this. You're comparing the microbiome communities associated with specific coral species across various locations. Between two islands, how do the microbes on corals from one island differ from those on another island? Within islands, within a single island, how do these microbes on corals differ? Sites differ, excuse me, on corals from different sites compare. Now, you could do it within sites at a specific site. How consistent are the microbe communities among individual corals? And by analyzing and comparing these, researchers can actually determine how geographic distance and local environmental factors influence the composition of coral associated microbiome communities. For instance, the greater the dissimilarity between the islands compared to within sites suggests that geographic separation plays a significant role in shaping these communities. Discussion and key findings. This study directly supports the need for better understanding and conservation of microbiome communities in local reefs. The Terra expedition conducted extensive cross-ocean sampling of coral reefs, revealing an unprecedented level of microbiome diversity across coral reefs, fish, and plankton. The sequencing efforts identified over 500,000 bacterial archaea ASVs, representing up to 20% of the estimated global microbiome diversity, suggesting that Earth's microbiome diversity is significantly underestimated. Microbiome communities were distinct across different biomes, with plankton exhibiting the highest diversity. This contradicts earlier studies, but aligns with the findings from multi-reef studies in Australia. Within coral microbiomes, diversity patterns did not follow expected geographic trends. Different coral morphotypes showed varying diversities across islands, but no single environmental factor could universally explain these variations. The microbiomes of corals did not align with known biogeographic regions of their hosts, indicating that environmental conditions and their host-specific factors, health, mucus production, played a greater role in shaping their microbiome community than historical or geographical influence. Lastly, geographic distance was the strongest driver of microbiome community composition, followed by environmental factors. However, environmental properties explained a smaller proportion of variance in coral reef microbiomes, 4 to 11%, compared to the plankton microbiome community, 29 to 30. Despite some similarities, coral microbiomes vary even within sites, and no universal microbiome taxa were found across all samples. And for questions, just please take a moment and pause this slide and go over and see if anything interests you. Then lastly, the references from the 3 studies that I pulled from